So what is the intervertebral disc? This was briefly already mentioned in the video that you saw before. So the intervertebral disc lies between two vertebrae within your spine, and it's like a spongy thing to dampen your walking, and it gives you the mobility within your spine. And then the intervertebral disc itself is made up of different regions. So it's like, I think you all had a jam donut in your life. So it has like an outer structure that is really solid and keeps it all together, which is called the annulus fibrosus. And then it has the inner bit, which is more gel-like, and it's called the nucleus pulposus. So disc degeneration is very multifactorial as osteoarthritis is. And you will hear from Paola a very different aspect of it with catabolism and how this whole degeneration cycle works. However, I will solely focus on bacteria today. So we are built of around 30 trillion cells. And within us and on us, we have around 38 trillion cells of bacterial cells. And they are often within and on us as like in sense of a microbiome. And what is this microbiome? So the microbiome is just a title for like the bacteria, fungi, viruses, and their genes, and they naturally live in and on our bodies. So depending on where they are localized, they can be classified into gut microbiome or the respiratory microbiome or the skin microbiome. And as you've read or seen in like various papers is that, for example, the gut microbiome is very dependent on, like, it can, for example, lead or is correlated with depression depending on how your bacteria, like what bacteria species you have. But they do live in symbiosis, so that means for example, the digestion, we do need bacteria, right? But there are also organs within our body that are considered as immune privileged. And immune privileged organs, they need to protect the structures, so there is no immune system present because the immune system might cause damage once it reacts. And those, for example, are the brain and the eye. But another organ considered to be immune privileged is the intervertebral disc. So the topic bacteria within the intervertebral disc is therefore very controversial. And the question of my PhD project was, are there bacteria in the disc? And if so, where in the disc are they located? And so to approach this, we obtain samples directly from the hospital, from patients. So when someone, for example, has low back pain that, like, for example, the disc is not intact, but the jam came out and pushes, for example, on the nerves, it's very painful. And so they remove a part of the intervertebral disc. And so we do get a part of the intervertebral disc, obviously from patients that gave consent. And as I work in the UK, from the NHS. And so there are different methods that can be used to look at the presence of bacteria. One of them is 16S sequencing. So 16S sequencing is when you get your samples and you snap freeze them, and then you look whether 16S is present. So what is 16S? 16S is an rRNA subunit present in all the bacteria, and it has constant regions and variable regions. Now, the constant regions are used to copy it up, that we have tons of it, and then the variable regions are used to identify which species it is. Another method used is microbial culture. Now, microbial culture is you take your sample and then you scrape it through with a blade and you smear it out on a plate where the bacteria can grow. Now, the bacteria, various bacteria species have different conditions how they want to grow. So, for example, we then need to have a tank with a normal oxygen condition and one with a lower oxygen condition, because as the intervertebral disc, for example, is an avascular tissue, the oxygen content is really low. So we have around 2 to 5% oxygen within the disc. And for example, C. acnes, one bacteria that is very often mentioned, as a natural habitat in the intervertebral disc needs to be grown at low oxygen and for up to 14 days because it's very slow growing. So the problems with both of those methods is they can't tell us whether there is bacteria in or on the sample, but they fail to really tell us whether it's contamination from when they took the sample out or whether it's true in vivo disc bacteria. So therefore, we performed immunohistochemistry. So for immunohistochemistry, when you receive the sample, you first cross-link it to fix it so that you kind of have a solid block and nothing within the tissue changes anymore. You then run it through a tissue processor and embed it in a blocks of wax. And this is kind of then just a piece of bread that you have, and that you can then cut and mount on slides. Just, we don't have as thick of a slide as bread, but it's 4 micrometers thin. And with that, you then can perform staining. Now, for example, if we stain for bacteria, as you might know from lectures that you had in the undergrads, gram-positive and gram-negative bacteria have different compositions of cell membranes. For example, the gram-positive, they have a monolipid layer with peptidoglycans on the outside. And gram-negative, they have a double lipid layer and peptidoglycans in the middle, whereas on the outside, they have lipopolysaccharides. So, okay, I need to just go forward and forward and forward here. So what we do is we add a primary antibody to our section, which then binds to a specific site of this membrane, in this case, lipoteichoic acid. We then add a secondary antibody, which specifically binds to its primary antibody. So you have a system that is very specific on binding one to each other. Okay. Yeah. On binding one to each other. And at the end, when you have the secondary antibody that is bound, you add DAB, which has a chemical reaction that turns the blue into brown. Now, our study looked at C. acnes. So therefore, we performed immunohistochemical staining only on non-herniated disc samples. So that means the herniated disc sample, as you can see here, is the one on the bottom, where the blue part, the nucleus pulposus, squishes out. The non-herniated ones on the top, that's a bulged disc. So it has a deformation of the annulus fibrosus. However, the nucleus pulposus is still inside and can therefore be considered as still sterile. So we only took those to not have contamination for the surrounding tissues. And we performed the initial staining on 95 samples. We did not only stain for C. acnes, but we also stain for receptors. So the toll-like receptors or pattern recognition receptors, the whole thing of toll-like receptors comes from the immune cells, and they can recognize, for example, peptidoglycans or lipopolysaccharides, and then trigger a downstream response, which leads to the expression of more pro-inflammatory cytokines. We also looked at the knot domain, and this was done the first time in the disc, and we detected it, and this is similar as the knot, it's just not on the cell membrane, but within the cell. And they have previously been detected in immune cells and epithelial cells, but not within the disc, as said. So here you can see some of the staining. So the brown bit is where, for example, here we do have C. acnes present, and the blue is just the nuclei stained with hematoxylin. So if you see a blue cell like you see in the upper part, that means that this is kind of a negative cell, immunonegative cell, and the brown ones are immunopositive cells. Now, to see whether we have unspecific staining, you can see in the top row the IgG and IgM controls. Those are just that you use an isotype, so the same building structure as your other antibody has, and you add it instead of your antibody to see whether you have non-specific binding through the form the antibody has. You can see here that C. acnes, so this is the bacteria that we looked at, was only detected within the cells and not as a biofilm in the tissue, which made us a bit curious, and we still don't know why, but we are looking into that right now. So how do we quantify what we stained for? So previously, manual counting was done of 200 cells. And as you can see here, it's pretty hard to quickly count through and say, OK, how many cells are what? So therefore, I apologize for my slides. They did not look like that before. They are differently formatted. So manual counting can be very subjective. It's not very reproducible, and it's very, very time intensive. So what we therefore did, together with my colleague in the back in blue, Maria Paula, yes, you, I contacted her because I wanted to do this as an automatic process and not manually. So we now have a Git book online that can help biologists to go through this process. So what we do is, so what you need to do it is just solely a slide scanner. So you feed your slides into the slide scanner, and then what results out of this is a slide scanning ID, and so the slide scan image for each slide, obviously. Then there is a software called QPath, which was created by Pete Bankhead for more looking at pathological features, for example, in cancer and so on. And he developed that, and we use it for our staining. So what we do first is we create a project. We create a project because we want to do batch analysis, meaning that we want to have the same settings for all the slides within one antibody. If you're familiar with microscopy, you know that if you do fluorescent staining, you will have multiple channels. Now, if you image with bright field, it's all on one channel and flat. So with estimating the staining vectors, this is a plugin that distinguishes then between the brown and the blue, which is very important for our later on process. So what we then do is we create a training image. So within all our slides in the project, we just select random regions that vary a lot and then put them together in a training image. We then perform the cell detection, which is also an automatic plugin. And as you can see, not all of the detected things are cells. And there is where the problem comes in. So this software would have an automatic detection for immunopositive cells. However, since the disc tissue is very low cellular, it detects tissue artifacts. And so to avoid that we have, so this is just how it looks like in the system, to avoid that we have wrong stain or wrong detections, we train our system with an object classifier. So we tell the system which one is a negative cell, which one is a positive cell, and which one is a tissue artifact. And then at the end, we also create a tissue detection. So either we can do that automatic by just setting a threshold, looking at the pixel, or we can do it manually, and this is how it looks like. So for example, this is a human sample that we had, which is solely NP tissue. And here you can see that we distinguish within the different regions within the tissue, because obviously sometimes it's very interesting to see in which region we have what, and are they even within all of the cells. So this is the whole process, how the script then runs. So within the script, as I said, you create a project, then you run it. It detects the tissue if you do it with automatic tissue detection. It then removes the edge effect, because if you perform staining, you usually have like a bit an accumulation of stain on the side. And then it runs the cell detection through and the object classifier, and then results in a result folder containing text files. So this is just to show you what it looks like. So you can see here the different cells. In pink is the negative, and in green the positive cells. Yeah. So As said, it results just in a text file giving us the negative and positive cells, but not yet the positivity rate. And there was the point where I was struggling, because if you have, I have 95 samples for one antibody, but I have multiple antibodies. And to then get it out of each text file would be very time intensive. So that's when I contacted Maria Paula, who then wrote the script on Python, which actually does take the text files and then calculates the positivity rate from the immunonegative and immunopositive cells. But it does not only do that, it also takes, remember that I said when we scan it, we have this slide scan ID, which we take for the whole analysis, but it also then links it up with our sample ID, so we can perform the whole analysis blinded and link it up in the end, and we get a nice file telling us which antibody, or the positivity rate for each sample. And then at the end, I used the Access database to link them all up together so that we can have like the different factors, because we're sometimes not only interested in one antibody, but the correlation between them. So for example, we also use the degenerative score, which we've done in histology, the age and the gender of the patients. So what you can see here is a Pearson correlation matrix with the p-values. And we can see that within our study, we found a correlation between the positivity rate of C. acnes and the degenerative score of the tissue. We also found a correlation between C. acnes and NOT2, which is the intracellular receptor, which kind of makes sense because the bacteria are internalized, right? We also found a correlation between TLR2 and NOT2 and TLR4, as well as the both toll-like receptors. We then looked at the different degenerative grades and the different factors. So we saw that C. acnes had an increase with degeneration, so the NOT2, TLR2, and TLR4. And this aligns, obviously, except the C. acnes, because this has never been done, and the NOT2, but the TLR2 has been associated with degeneration within the tissue. We then also looked at C. acnes within the different age groups, and we did see, first of all, that C. acnes was in all the samples, which aligns with what Dr. Rasha Sukharan said, that the disc might just harbor a natural microbiome, so that C. acnes could be a natural habitat of the disc. And since we did find it in all of the disc and in all the different degenerative states, this might be the case. So Modic changes have been a very rising topic within the disc field, and Modic changes are vertebral bone marrow lesions that have been associated with infection of the disc as being one of the possible etiologies. And we did see, so same as the group from Zurich, which is really the pioneers in that field, that we did have two different groups of Modic changes. I mean, there are Modic changes type 1 and Modic changes type 2, but looking at Modic changes type 1, we did see a group of higher bacteria present and a group with lower presence of bacteria, and this aligns with the study done by Irina Hekli. We don't know why, though, yet. So in conclusion, I would like to say that C. acnes was detected in all the samples, that NOT2 is expressed in NP cells, and its positivity rate correlates with the positivity rate of C. acnes, and that upcoming staining for catabolic markers will hopefully help us to really understand whether C. acnes triggers a catabolic response within the disc or whether it's just naturally there. Yeah, with that, I would like to thank my supervisors and the whole Disc for All consortium, and I'm very open to questions.
Thank you, Andrea, for this very nice talk. So we have quite a lot of minutes, like 50 minutes for questions. Someone wants to start?
So what's the name of object classifier here?
The object classifiers, the name.
Yes.
What do you mean with the name?
So you used the object classifier, no?
Yeah, which is just a simple plugin within QPath. And you train it.
Okay.
Or what, sorry, I don't quite get what you mean.
So can you go back to slide?
Yes.
So you used the object classifier, no?
Yeah, but this object classifier, so QPath is an open access software.
Okay. So which I
And Pete Bankhead, so he programmed it for, wait a second, I need to see what it is. You mean this part, right?
yes.
So the algorithm behind, I don't know, because I'm a biologist. I just use the program. It's JavaScript based, and he just done different plugins that you can use, which I encourage all of you to do some stuff for biologists that has a simple interface that we can use to click on and then train an object classifier, because that's all I do.
Okay. So you explained about some correlation, no? Before previous slides.
Mm-hmm. You just say stop. Are you sure it's before and not after?
I'm not sure, but you explained about some correlation between features.
Oh yeah, but that's later, sorry.
Yes.
You mean the matrix, right?
Yes, here. Can you explain this more?
The statistical analysis.
Yes.
So it's a Pearson correlation test, and if it has a value between 0.05, which is statistically significant, you can then see, so the R value, I don't have the R value here, which would be, so the Pearson correlation would give you the R value, right, and the P value. And what it would give you is if they somehow correlate with each other. So for example, the age and the positivity rate here, right, which is a pretty big number, does not correlate with each other, whilst if you go below 0.05,
Okay.
or do you want me to explain the statistical test itself? Because
So its maximum value is 1, that correlation maximum value.
this is not the you don't get a value of correlation equals 1. No, that's not it. So what you just look at is whether you have a correlation. It doesn't tell you. The R value would tell you how much they correlate, like how much they align with each other. This value here just tells us whether they somehow correlate with each other in terms of if I have a bigger value here, I have a bigger value here. But this is a P value, so like a significance value, not
Okay.
a 1 value.
Okay, thank you.
Thank you, Kari. Any more questions?
Yeah, thank you, Andrea. I think that's super nice because you're basically, so this basically then opens totally incipient and still new field, which is coupling all what Christina has shown was a further problem of bacteria. So we usually see bacteria as we have an infection, we're sick, and now I guess you're suggesting that then there is a subclinical or even normal form of bacterial colonization then in those tissues that are here, whether we like it or not. And so if I understand correctly, all but is subjacent to this work, a final conclusion you might have, a final hypothesis you might have, is that we have those bacteria in the disc, it's hypoxia, so they're developing very slowly, and then so they would then contribute to the natural then aging of the disc through basically then the biological pathways you have identified. Would that be your final word about this work?
I think I need to do more analysis on the bovine stuff that I have done. which we looked at. We looked at bovine discs that we infected with bacteria to see what we get out of this, to see whether bacteria have an influence or not, because we detected C. acnes also in the bovine discs. So I would say we do have a natural microbiome. I would agree with that. What its influence is on degeneration of the disc, I wouldn't say anything yet, because I think more analysis needs to be done before jumping to any conclusion, because I would like to first see some facts before stepping out and saying something, to be honest. Because in our study, like, it has, as you can see here, it does correlate with degeneration, but you can see the graph too, right? It's not like a straight line that is very nice. So it can well be that it's like, you know, together with different factors that it could maybe enhance. And also bacteria can be within the cells and be just in a hybrid state, that they just are there, but they don't do nothing because they can just sleep in that sense. And that they like it within the disc. I mean, C. acnes is super anaerobic, so I think, yeah.
Just a comment on that as well. Remember, there's been many clinical trials on the use of antibiotics for this degeneration, which correlates.
50-50, yeah? They work 50-50. But also, if you have antibiotic treatment and the bacteria is within the cell, it might not even affect it, you know, so it might just be, so it might also depend on the infection you have.
But the interesting thing is as well, as you said, you didn't actually see them getting in or out of the cell, right? So you didn't see them within the tissue.
No, but we did, someone else has done the study too, but we also looked at it ourselves. We had disc cells in monolayer and added the bacteria to see whether they even get like, within the cell, and they did. But the question is just also, is it something that comes from the cells that they engulf the bacteria, or is it something from the bacteria that this process is just, it's not known yet what exactly happens. It has been shown that they can like macrophage, so NP cells can be macrophage-like, and that they can do phagocytosis of things, but the process how and why exactly is not yet, researched.
Okay. Well, I would have a follow-up question, so this might be a little bit critical, but interesting in my opinion. So we did like now for 70 years intervertebral disc studies on intervertebral disc cells, right?
Yes.
But based on your completely new findings, those might be biased in a certain point, in the sense that we didn't actually maybe have all the variables yet to be able to find degeneration, because one critical value is what you're pointing out now, which wasn't known. So how would you kind of estimate the impact of this for the research field itself?
That's a good question. I think it is of importance, but I also think that there are so many different factors that play within each other that I think it's good to know, it's good to have in the back of your mind. But I said, I don't think it's the driver to disc degeneration. So it might have an influence. I don't think it's the new thing and we can now cure it. Not at all.
Okay. Are there some more questions?
Yeah, actually, the fact that so you're able to establish then a relationship between Modic change type 1 and then bacterial colonization. So actually, you're not looking at the same level within the intervertebral disc, because here you're looking at nucleus pulposus cells. Is that so? So then Modic changes will happen and at the end plate level.
But so it is associated with like whole infection of the disc, not just end plate.
Right, but then the question is, and then this is not based on what you're showing, and I'm asking for your own interpretation of things. But so would that be possible then there is first, so a biological disturbance at the end plate. So the end plate is the interface between the intervertebral disc and the vascularized vertebra, okay? There's first a disturbance here that would make somehow the barrier leaky. and then which would then favor this migration of the
I mean,
of the bacteria.
it
Which
could
means
be,
that
but
in
we
very
do
young
have-
disc, you would
we-
not see anything.
We do have from dead people, we do have cadavers in our data bank. So we did look at those, which did not have any signs of degeneration of the disc. And we did find the bacteria in those too. We did find them in the bovine discs, the cyagnus, which have still very intact, because those are very young animals. They have intact end plates, and I mean, I can see that on my histology slide that there everything is intact. And we still find the bacteria. So I don't know, more studies need to be done on the porosity of the end plate too, to really be able to see when they get in. I mean, it would also be interesting to see if they are maybe already in like neonates. Like, I don't know, we don't know. And yeah.
We would still have time for a question if someone wants to raise their hand now. What's still with time? Okay, Jérôme.
Now, just to comment on what Roger said, so the antibiotic treatments, most of antibiotics have electrical charges. And actually, it's super controversial whether they can really penetrate into the intervertebral disc, because what do we have in the intervertebral disc? Protoglycans that are negatively charged. So here there is some physics involved into that in order also then to correctly interpret whether this antibiotic can effectively then reach the bacteria within the disc.
Maybe I will have a last question. I was just curious whether you saw this bacteria always throughout the samples, or if it was somehow in clusters. You know, if the
I can show you a slide to that, which is in the bovine, not in the human. Wait a second, I need to see what it is. Not in the bovine, but in the not in the human, but in the bovine. So you can see that the inner part is more green, which means that those would be the positive cells, and the pink ones are the negative cell, immunonegative cells. So this is cyagnus in the bovine disc. And we can see that somehow the outer annulus fibrosus, where we have a higher oxygen concentration, seems to have less cyagnus present than the inner stuff. However, this is like, I didn't do the full analysis. This is just what I have yet seen on the images. But this could indicate that we do have a higher bacterial load within the NP than within the outer AF.
Okay, interesting. Someone else? Because if not, I would close this session to be able to start the next one punctually. So let's thank the speaker again. Really great talk, Andrea.